Morning, good afternoon, good evening all. I'm Monica Setia, Associate Country Director for India at the Lakshmi, Mittal and Family South Asia Institute, Harvard University. A warm welcome to all our audience members, esteemed panelists, team members at the Mittal Institute, and representatives of the co-organizing entities for the webinar, Lessons from the Global South, How to Develop Life-Saving Vaccines for All. As you might be aware, the webinar is being organized jointly by the David Rockefeller Center for Latin American Studies at Harvard University, Harvard T.H. Chan's School of Public Health, Lakshmi Mittal and Family South Asia Institute, and Lancet Citizens Commission on Reimagining India's Health System. I'm not going to take too much of your time and just quickly lay the ground for the next 2 hours that we all will be spending together. After a brief introduction by our Executive Director, Hitesh Hathi, whom I'm going to introduce very shortly, Professor David Williams, our session chair, will give opening remarks, which will be followed by presentations by 3 panelists, Veronica Vargas, Gerardo Julian, and Regis Ella. Our discussant, Osman Wahid, will spend the next 20 minutes or so engaging with panelists in a conversation which will be followed by our audience Q&A for a good 25 minutes, and then we'll have closing remarks by the session chair and a vote of thanks by Hitesh. We request all audience members to use the Q&A box feature in Zoom to post their questions. The discussant will call upon you to address your question, at which point you will be requested to unmute yourself and come on the screen if you like to ask your question. If you prefer not to come on the screen, please put a note regarding that along with your question, and we'll be happy to address your question by reading the text that you would have put in the Q&A box. And without further delay, let me introduce Hitesh Hathi, Executive Director of the Lakshmi Mittal and Family South Asia Institute. Hitesh has a long and rich experience in leadership and management and a deep lifelong engagement with South Asia. He worked as a producer for one of the Boston's leading NPR stations for over 20 years, with his last assignment there as an executive producer of the show Radio Boston. He also has a long connection with Harvard University, having received his Master's of Arts degree in Sanskrit and Indian Studies from Harvard and being a PhD candidate in the Department of South Asian Studies at the university. Hitesh has also been an assistant and acting resident dean at Harvard's Cabot House. It's a great pleasure for all of us here at the Mittal Institute to be working with him. I'll hand over to him to take forward the conversation from here. Thank you so much, Hitesh, and a warm welcome again to everyone.
Monica, thank you very much. I want to just welcome our distinguished panel of guests. It's really an honor to have you here, including some old friends, and welcome to everyone who is listening online. In a good, whatever part of the day you're in, and we know you're from all over the world, so morning, afternoon, evening, as Monica says. So I just want to say that for the Mittal Institute, it's really a deep honor to be involved with this session, and also to be involved with so many distinguished units within Harvard and outside of Harvard who are part of not just this particular session we're doing, but thinking at large about how to work in productive ways across borders, across all kinds of lines and divisions. The Mittal Institute at Harvard is dedicated towards working across every school, every departmental boundary at Harvard. So all of the projects we do are multidisciplinary, from the Graduate School of Public Health to the medical school, to the School of Education, to the Divinity School, School of Design. We have scholars working with us on multiple projects on from every part of the university. And within South Asia, we are dedicated to working across every border and every kind of division and siloing that happens for productive ends. And that's particularly fit for this session, where in fact we're talking about how to achieve maximum public good internationally, and the role that the Global South can play in it. So I'm very honored to welcome all of you here. I won't talk very long because we have a very distinguished panel, and most of all, we have a very distinguished leader of that panel. And it's my pleasure to be introducing David R. Wilkins, who will take us forward and will give us more of a context for what we're doing today. David R. Wilkins is the Norman Professor of Public Health and Chair of the Department of Social and Behavioral Sciences here at the Harvard Chan School of Public Health. He's also professor of African and African American Studies and of sociology at Harvard. He's also the author of 500 plus scientific papers. His research has addressed the complex ways in which race, socioeconomic status, racism, stress, health behaviors, and religious involvement can affect health. He's an elected member of the National Academy of Medicine, the American Academy of Arts and Sciences, and the National Academy of Sciences. David, please take over. And for those listening, I will take myself off screen because I want the focus to be on the presentations.
Thank you so very much for your kind words of introduction, and welcome to the panel. Let me set the context of what we're doing today. The COVID-19 pandemic has been the most devastating public health emergency of our time, affecting billions of people around the world. It has also triggered considerable scientific innovation, including the development and clinical trial of over 200 vaccines worldwide. Among them, the mRNA vaccines involved the use of groundbreaking technology and played an invaluable role in addressing the pandemic in the US and other rich countries in the world. At the same time, there were remarkable achievements of other vaccines from the global South, including India and Cuba, that repurposed existing safe vaccines like those targeting hepatitis B or rabies. These vaccines offered important benefits such as affordability, ease of storage, and well-established safety profiles. In June of 2022, I traveled to Cuba as part of an international delegation of scientists from the United States, the Caribbean, and Africa. We were on a 3-day fact-finding mission to better understand Cuba's COVID-19 vaccine development and vaccination efforts. It was the first time in 5 years that a scientific delegation with significant US presence had engaged in discussions with medical researchers in Cuba. Together, delegation members represented a broad range of expertise in public health systems, infectious disease, biotechnology, and vaccine development. And I learned a lot from that trip, and I want you to learn all what I learned and more. So this webinar will delve into the main challenges of repurposed vaccines that they face, how those challenges were addressed, and what lessons we can learn not only from Cuba, but also from India for future pandemics and neglected vaccines as we work together to ensure equitable access to life-saving technologies, including vaccines for all in our world. Now it is my privilege to introduce the 3 panelists and a discussant who we will hear from. Veronica Vargas is an economist with expertise in global health, health economics, and pharmaceuticals, and she boasts over 25 years of experience in academia and international organizations like the World Bank and the Pan American Health Organization. Currently, a research associate at the Lakshmi, Mittal, and Family South Asian Institute. She focuses on fostering collaboration in vaccine and therapeutic innovation, particularly in the context of COVID-19 in the Global South. Dr. Vargas also serves on the scientific panel of the International Health Economics Association and co-founded the Global Health, Biodiversity, and Therapeutics Innovation Consortium to promote pharmaceutical innovation and access. Our second panelist is Dr. Geraldo Guillen. He is the Director of Biomedical Research at the Center for Genetic Engineering and Biotechnology in Havana, Cuba. He received his bachelor's degree in chemistry and a master's in chemistry in 1986 from Mechinov University, Odessa, and his doctorate in biological sciences in 1995 from the University of Havana. His research focuses on prophylactic and therapeutic vaccine development against hepatitis, dengue, meningitis, whooping cough, cancer, COVID-19, as well as in drug development. Dr. Guillen is a professor at the University of Havana and the Latin American School of Medicine. He's also a distinguished professor at the University of Science and Technology of China and a member of the Cuban and Tijuana Academy of Sciences. He has published more than 65 scientific articles and has developed more than 62 patents. He was awarded a gold medal for the Abdala vaccine patent granted by the World Intellectual Property Office. Our third panelist is Dr. Raches Ella. He's the Chief Development Officer at the Bharat Biotech. He leads Bharat's Global Product Development Division, focused on discovery and clinical development for vaccines and therapeutics. He also oversees medical safety and regulatory support for the entire pipeline and external science and innovation. He's a physician by training with an MS in clinical research from Emory University. Following his postdoctoral fellowship at Johns Hopkins, he also completed his MBA at Harvard Business School. And then after the three panelists have spoken, we will listen to a discussant, Osman Khalid Wahid, the CEO of Feroz Sons, I hope I'm pronouncing that correctly, Laboratories Limited. He joined the company after completing his undergraduate studies at Harvard University in 1993. In response to local health challenges, this company has expanded its portfolio through strategic partnerships with global entities like Boston Scientific and Gilead Sciences, Inc. During COVID, the company worked closely with Gilead to deliver the COVID-19 therapeutic remdesivir to low and middle income countries. Wahid is a trustee of the Lahore University of Management Sciences, Pakistan's leading multidisciplinary university, and co-founder of the Lahore Biennale Foundation, a nonprofit that supports public art. So you have heard about the distinguished panelists and discussants we will hear from. And now I will turn to the panelists to present to us. Dr. Vargas, I apologize. We are having some technical difficulties. Why don't I go ahead with the second panelist and we'll come back to Veronica. So at this point, let me call on Dr. Gerardo Guillen.
So I will share my screen. So thank you for the opportunity to share the Cuban experience with you. I will talk about the development of mainly of Abdala vaccine. I will mention all the Cuban vaccines in general, because in Cuba we were able to develop 5 vaccines, 3 already registered and 2 that will be registered soon. And if there is a little time at the end, I will talk also about our challenge for the future and a couple of slides about dengue vaccine that is very important for our region, for tropical and subtropical area. So talking about the vaccine and the CIGB, this is the Center for Genetic Engineering and Biotechnology in Havana. It was very important, the large experience that our center have in more than 30 years, developing different recombinant proteins. That you see in the screens are the different recombinant proteins, the different biological products that we develop and we produce from many years ago. The most important is also the pipeline. We have around 30 different projects under development, more than half of them are based on recombinant vaccine, using this platform for production, and also E. coli and synthetic peptide. 8 out of the 30 projects that we have in our pipeline are prophylactic and therapeutic vaccines. The most important for the development of COVID vaccine was the experience we have with hepatitis B vaccine that was developed by our center since year 91 and was introduced in the health system in year 92. So this vaccine is produced in yeast, in pique pastori yeast, at 3 cubic meter fermentation capacity. And more recently, in the year 2015, we developed the therapeutic vaccine against chronic hepatitis B using the same platform for the production of hepatitis B surface antigen. So this was very important because we used the same platform, the same technology, the same host, and the same production facility to develop the Abdala vaccine that has been used in around 80% of the Cuban population. Abdala vaccine was based on RBD, receptor binding domain, expressed in yeast, in pique pastori yeast, purified to more than 98% purity, and adjuvated to aluminum hydroxide. So it's a very simple vaccine with very high purity. The scale up was in parallel to the clinical trial, up to 3 cubic meter fermentation. We patent this construction, this vaccine, and we publish also the vaccine, the development of the vaccine. You see in the screen the information available at that time about the sequence of the virus, the sequence of the spike protein of the receptor binding domain, the information about the receptor, the ACE2 receptor, the information about the glycosylation of the protein was very important to refine and decide our strategy to develop this vaccine. It was very important during all the time the open access publication, or the publisher have open access all the publication related to COVID, and was very important also the preprint, because every lab published, you are preparing in advance, and that was very important to share that information for the vaccine development. So we decided to use this platform, yeast platform, recombinant protein, because it was the one that we have experience and the one that we have production capacity, and because this platform was able to glycosylate the receptor binding domain, that was very important based on the information available. So the advantage that pique pastori also have a high glycosylation rate, that is very important also for immunogenicity, for stability of the protein. Around 80% of the sugar content is mannose, that is immunopotentiatory cells, and all this was very important in our decision about the strategy. So, we conducted very rigorous clinical trials, phase 1-2 clinical trial. We were comparing 2 schedules, a shorter schedule, only 2 weeks apart, 3 doses, only 2 weeks apart, and a longer schedule, 1 month apart between those. And we also were testing 2 different formulations, one with 25 micrograms of the RGD antigen, and another one with 50 micrograms. So they were 6 different arms in phase 1 clinical trial, and 3 different arms in the phase 2 clinical trial, because we selected the shorter schedule and the higher dose to continue ahead with our vaccine. All the trials were randomized, placebo-controlled, and we included more than 84,000 volunteers in phase 3 clinical trial with a shorter schedule, because the pandemic was important to select the shorter schedule, and the higher dose, 50 micrograms, that was more immunogenic. They were included 18 different clinical sites in three different provinces, and the support of the local government, the national government, the confidence of the population in our health system and in Cuban science was very important, because we were able to include all the volunteers in only 2 weeks. So at the time of the administration of the second dose, already all the volunteers were included, the 48,000 volunteers with the first dose already administered. So this allowed us to conduct very fast the clinical trial, and we were taking decision in parallel during the clinical trial. For example, at the time that we have the results of the shorter scale, we decided to go to the next trial with the shorter scale, not waiting for the longer scale, because shorter scale was the more convenient for the pandemic, and based on the result was immunogenic enough. So the efficacy of the vaccine was 92%, was very rigorous follow-up with the double-blind trial until we accumulate the number of cases enough to test the efficacy. We didn't open the cut, and it's important to mention that we excluded for all the analysis the volunteers that were positive to coronavirus in time 0. So that was very rigorous determination of the efficacy of the vaccine. It was 92 efficacy, and it's also important to mention that this efficacy trial, the phase 3 clinical trial, was conducted at the time that the main strain that was on circulation was beta strain. And you see in this graphic, the phase 1-2 were conducted at the time that this 614 strain was circulating, but at the time of phase 3 trial was mainly beta strain in red in the graph. And the effectiveness study were conducted at the time of delta strain that you see in yellow in the graph. So that is very important, because our vaccine was a few months later that mRNA vaccine and Sputnik and adenoviral vaccine for AstraZeneca. So they tested the vaccine at the time of alpha and 614 strain on circulation, but we tested our vaccine at the time that beta strain was circulating in Cuba, and beta strain was the most resistant strain to neutralization. So that I mentioned was very rigorous clinical trial. So the effectiveness of the vaccine in open population in Havana was more than 90% also. Again, hospitalization, severe illness, and death. That was very important achievement. And another important point.
Seeing all of the notes here?
Yes, I don't see this. Yes, my note, no, that my notes are not there. Yes,
Okay.
yes.
Are you hearing okay? Are you hearing me?
Yes, Gerardo, we can hear you. Please continue.
Yes, we can hear.
Yes, we can hear.
So that you see in the screen is the how was the different epidemiology of the COVID in Cuba. So in black that you see the main peak was the Delta infection in Cuba that was a very high, and when we reached more than 60% of the people immunized, the epidemic started to go down. And at the time of Omicron, that is the small peak that you see in black in the screen, at the time of Omicron, we have more than 80% of the population immunized, and we have more than half of the population already with the booster. So the effort and the universality of our health system, fast introduction, the fast administration, and the high coverage of the vaccine. And as you see in the screen, the behavior of the epidemic in Cuba was completely different than happened in the world. So Omicron wave was higher than Delta wave in the world in general, but in Cuba was 10 times lower, both mortality and epidemic, thanks to the high coverage and the efficacy of the vaccine. So that is not only the capacity to develop the vaccine, but the capacity to have a strong and universal system with the capacity to achieve very high coverage of the vaccine in very short time. So, and we also conducted a clinical trial in children, phase 1 and phase 2 clinical trial, and then we conducted a brick study where we compared the neutralization capacity from the cohort of people of younger population, less than 21 years, 19 to 21 years old from the efficacy trial in adult population, and the children from 2-year-old to 18-year-old. So we compare the neutralization capacity of the sera for the younger cohort of population from the adult population in the efficacy trial with the children population. And as you see in the screen, there was very high correlate between antibody titer and neutralizing capacity of the sera, and that allowed us to correlate the efficacy of the adult population to children population and allow to register the vaccine for population older than 2 years. Another important advantage of the platform that we select, all the Cuban vaccines are based on recombinant platform. In the case of Abdala, that is the one that I explained, but in the case of Soberana, that was developed by Finland Institute and Center for Molecular Immunology, that is based also on RBD antigen, but using a mammalian system, CHO cell for production. So we are using a recombinant platform for all the Cuban vaccines. And very important advantage of this platform is the safety. So when you compare with other vaccines, we only have in our surveillance study from the National Health System, we only have mild side reaction and limited side reaction compared with RNA or adenoviral vaccines. And another important point to highlight in the development of our vaccine was the integration between different institutes. So many institutes contribute to the development for our vaccine. So for example, the Center for Immunoassay was responsible for all immunoassay, for all immunological assessment. The National Defense Civil Laboratory was the one that conducted the neutralization test together with the Center for Tropical Medicine, that is a reference laboratory from WHO, so that the integration between different systems was very important. The CRO, Sensei, was the one that monitored and conducted the clinical trial. So the cooperation and integration between different institutions and the support of our government, of our population, was very important. So the vaccine become proud of our nation. All the population contributes significant. We have not a movement against the vaccine. We have completely the opposite situation, a high confidence of the population on our health system and on our scientific institute. In the screen, I will not read this, but it's just to list available for everybody the main publication, the patent, and the main publications about the development of the vaccine and about phase 3 efficacy trial, effectiveness trial. But in this website here from our center, you can download many other publications that we have related to the vaccine. And I wanted to remark the experience of the CGB developing the vaccine that was very, very important. The more than 30 years developing the recombinant vaccine production and conducting also clinical trials, both in pediatric and adult population. The sample composition of the vaccine, the confidence of the platform that we have been using, that was very important for the regulatory bodies, and the safety of the vaccine, the efficacy and effectiveness in adult and pediatric population, and the neutralization capacity against different variants of concern. Abdala was capable to induce a strong immunological response after primary immunization and induce memory response after booster dose. The suitable administration schedule of 3 doses administered in a very short time, with only 1 week, only 2 weeks between doses, and that can be easily incorporated in vaccine campaigns or vaccine programs, especially during pandemic scenarios. We also developed nasal vaccine. That is very important for the future, because nasal immunization is the only that will be able to induce sterilized immunity and break transmission. So for this, we use the same LBD antigen together with nucleoparticle antigen from the hepatitis B virus, that this antigen contains more than 20% of RNA, so it's able to induce innate immunity, to activate top-layer receptors, and also to use B cell, that is 80% of the immunocyte that we have in circulation. So this antigen is able to activate and to use B cells as antigen presented cells. So it's also very simple and recombinant antigen composition of the vaccine. So the vaccine was able to induce both serum and nasal mucosal response, EGA, EGG, functional activity of this antibody that you see here in this slide is that the vaccine was also able to induce very high level of antibody in older population, with no significant difference between all elderly population and younger population. The antibody titers were even higher in elder population, as well as neutralization capacity of the serum, and that is another advantage of the mucosal immunization, that the elder population have a full capacity to develop immunological response, which is not different from the younger population when you use a mucosal route of immunization. And that is very important for respiratory diseases. That we show here a small trial that we conducted using a booster dose for one group with systemic vaccine, Abdala, and another group with a nasal vaccine. And that we achieved is that only nasal vaccine was able to induce tissue-specific memory cells. So mucosal is tissue-specific memory cells that was not possible to achieve with the systemic vaccine Abdala. You see the time 0 and time 1 vectors are located very close. It means that it's not significant difference between the marker of tissue-specific memory cells in this vaccine. But when we compare mucosal vaccine, you see here in yellow time 0 and in red after booster immunization, and you see the different markers that determined by qPCR, and was significant difference, significant induction of a marker of specific tissue-specific memory cells, so that only mucosal vaccine was able to induce strong mucosal response. So the remark for this part is that vaccine capable of enhancing mucosal immunity may be an adequate tool for exhibit advantages in induced immunity at the nasopharyngeal level, in addition to the systemic response, and elder population could obtain additional advantage of mucosal immunization. I wanted also to address, because the organizers were asking about the lesson that we learned for this epidemic, and the challenge that we faced during the development of the vaccine. And one important challenge for us was the lack of reference material, of standard, of animal model, region for cellular response, for production, even vials as syringes, was shortage of supply, and that was especially a challenge for a small country like Cuba that have not the capacity to produce all the supplies that we need. It was also difficult to publish the results. The results were now that for a South country it's not so easy to achieve the publication of our results in high impact journals, so it takes more time. Now everything is published. That was difficult, and it took time to have our paper accepted in different journals. We need to apply to different journals. So especially for Cuba, it's the main difficulty is the financial and economic blockade from the United States. We were not able even to buy oxygen at the time of the bigger incident of the disease in Cuba. United States forbid to export ventilators to Cuba. The same happened with all the supplies that we needed for the development of the vaccine. Another important challenge was the WHO acceptance of the vaccine. We are still dealing with WHO, so we sent our dossier to the WHO, but our vaccine was a few months later that Pfizer, Moderna, AstraZeneca, the fast track for WHO acceptance was not the same, was not the same urgency, and we still are dealing with that. So, and another important point is the available resources. So when companies receive billions of dollars for the development of the vaccine, I'm talking about the big pharma company, Cuba was barely able to invest $70 million for the development of our five vaccines. So, and the lesson for the future. So I think that the South understood very well that we must rely on our own effort and our own capacities to develop vaccines. We must increase South-South cooperation and technology transfer, and we must develop our research and production capacity. We must work for regulatory homologation within our country to allow fast introduction of our vaccine in different countries. We were able, for example, to provide our vaccine to Mexico, to Vietnam, to Venezuela, to Syria, to some Caribbean islands, and that was very important in topic in South-South cooperation. We have very high experience transferring technology to South country and developing our cooperation with different biotechnology. So if you allow me a couple of minutes, I wanted to mention another challenge that we are facing now.
We don't have much time, so maybe a couple minutes, then okay. we have to move on to the next panelist.
Okay, just 1 minute. Just to mention a challenge for the South, that is the development of dengue vaccine. So far, the available vaccine, the only available vaccine is from Takeda to dengue that was registered in the year 2021 in Mexico, and from last year was registered in many Latin American countries, and also was recommended by WHO. Dengue vaccine from Sanofi was already discontinued, and the vaccine developed by National Institute of Health will be submitted to the ANVISA, the Brazilian regulatory agency, at the end of this year. So these vaccines are live vaccine, and these vaccines are based on the induction of antibodies, neutralizing antibody against the virus. And the antibodies have the limitation that the same antibody that neutralize the virus have the capacity to cross-react between different dengue strains and amplify the disease. That is a big challenge, because with time, the antibodies wane, and the same vaccine that at the beginning can induce protection, later can induce an increase on hospitalization that happened with dengue vaccine. That is why another challenge is that the vaccine doesn't protect against all the virus, and that increases the risk for immune enhancement of the secondary infection after immunization with different strains of the virus. And that is why Cuba selected the recombinant platform for the production of the vaccine, and our vaccine is focusing only on cellular response and not in antibodies. So that allows us to have a very safe vaccine, a very safe platform that will allow to conduct faster clinical trials and faster introduction of the vaccine. And we are open for cooperation with other South countries for the development of this vaccine. So I will not read all this. I already mentioned the main points, and thank you very much for this opportunity to share our results and our experience. Thank you.
Thank you, Dr. Guillen, for the very comprehensive overview you've given us of both the challenges, but also the remarkable successes that Cuba has experienced in producing the COVID-19 vaccine, but also in producing vaccines in general. At this time, I'll continue with the panelists, and I will go to Dr. Ella next, and then we will have Dr. Vargas come in as the third speaker. So, yes.
Thank you for the introduction, Professor Williams. Allow me to share my screen.
Yes.
I hope it's visible in full screen.
It is visible in full screen. Thank you.
Fantastic. Dear panelists, all hope is not lost. I think the Global South is innovating vaccines, and I think the presentation before me was an example of innovative vaccines coming out of Cuba. Today, I'll be talking to you about some of the vaccines that come out of India that are very novel indeed. Before I get there, I'd just like to paint a picture of the Indian pharmaceutical industry. Many of you are probably Indian, so you probably are aware of this, so just bear with me. The Indian pharma industry as a whole supplies 40% of generic drugs. So these are small molecule drugs. Just imagine your Tylenol sitting on your shelf. The odds of that being manufactured in India is about 80%. 40% of generics to the US, 25% of generics to the UK. Generics, though, are not innovative, though. You essentially wait for these drugs to fall off a patent, and then you start developing me too or generic versions of these drugs. So there's not much innovation in there, but there is some amount of innovation for process optimization to scale that up. The second bucket in the Indian pharmaceutical landscape is the biopharma, the biopharmaceutical landscape. So these are essentially drugs that are mostly presented in a liquid presentation. So it's not a tablet, usually injected into your muscle. And predominantly in the Indian pharmaceutical space, this biopharmaceutical bucket contains vaccines. Overall, Indian vaccine manufacturers contribute to 50% of the world's supply of vaccines. And so we've made some remarkable achievements in the past. In the 90s and 2000s, the Indian pharmaceutical industry helped towards driving down the cost of HIV drugs by about 96%. So with economies of scale, these companies were able to cut these costs and ensure that they're affordable to beneficiaries, not in all parts of the world. We're talking about HIV, tuberculosis, malaria. So these are some very important contributions that the Indian generic pharma industry has provided. But the challenge remains with at least India is the percent of GDP spend that we spend on R&D. And as you can see by a few metrics that I've provided here on the screen, 0.7% of our GDP is only dedicated towards R&D. And if you compare it to our peers and our neighbors, we're off by a few several fold. So in terms of how we are to improve, how we are to innovate, I think our emphasis and focus on R&D and manufacturing needs to be improved. Coming back to that biopharma landscape, the drugs that are presented in a liquid presentation, it's a rapidly evolving industry here in India. As you can see here, we're more bottom heavy. So we have a huge amount of mid-sized pharma companies, and we also have a decent amount of vendors and suppliers that provide us with this industry. But as you can see here, we're kind of top light. So if you look at the small and mid-size and the academic and research institutes that contribute to licensed products or licensed vaccines, I can only count a few. So in that regard, our focus from the academic level is also still missing. So it's very hard for us manufacturers in India to develop our own products without this entire ecosystem being robust in nature. Coming to vaccines, I think you picked the right webinar. In terms of a return on investment, for every $1 spent on vaccines, you recruit $16 in costs related to healthcare, wages, and productivity. So hopefully vaccines are here to stay, and we'll be continuing to innovate. Now, before I get to innovation in the Global South, it's very important for me to introduce to you a consortium of vaccine manufacturers that predominantly belong to the Global South. This consortium is called DCVMN. I know that's a mouthful. It stands for Developing Countries Vaccine Manufacturers Network. As you can see here in the world map, these are highlighted by countries that are predominantly in LATAM, Africa, India, China, and South Korea. And all of these countries come together towards providing vaccines at scale. And the overall consortium of DCVMN contribute a fair amount of vaccines to Gavi. Now, this is a dated slide into 2016 that showcased here in the pie chart on the right that 60% of global vaccines, these are mostly for pediatric immunization programs, have been provided by this consortium. So companies that belong to the DCVMN have been contributing close to 60% of the world's need. And it's been about 8 years since this slide has been developed. I'm fairly certain that metric has improved to about 70-75%. And it all started off with the Indian, at least the DCVMN manufacturers, starting to work on generic, Me Too, or biosimilar-like vaccines. So we first started off with a very conventional vaccine that have been there for decades, meningitis, measles, rubella, TB vaccine, and whatnot. And we first started there with those generic approaches. And as we make our way down the list of these antigens that you see here, or pathogens that you see here, we're starting to now work on innovative vaccines, or vaccines that multinationals have a high market share in, or complicated vaccines for several reasons. So that transition is currently ensued with regards to innovating with these vaccines and different platforms. And fairly and surely, I'm pretty sure that we will start, work has already started with these other novel pathogens that are highlighted in this green box. And lastly, the Global South diseases are, I think, a Global South problem. I'm finding it harder and harder to see multinationals work on these problems. And when I say global south diseases, I mean diseases that pertain to dengue, chikungunya, malaria, tuberculosis, HIV, or typhoid. If you look at any other large pharmaceutical company out there outside of the global south, they are not probably working on these diseases. It is not in their pipeline. It's not on their website. It's not probably a sound business model for them for understandable reasons. So I think the impetus is for us, for this consortium, at least, of developing countries, manufacturers, to start looking at these diseases and trying to find solutions for it. With that aspect, I'll just pivot to COVID. And the company I work for, Bharat Biotech, which is headquartered in India, where by volumes we're the 5th largest vaccine manufacturer in the world. And with COVID, we were able to collaborate with 4 different institutions to work on several vaccines. One of the first vaccines we licensed was the intranasal COVID-19 vaccine in collaboration with the Washington University. The whole promise of this vaccine was you administer the vaccine in the nose through a spray or a drop, and in essence, it induces an antibody responses in your nose, thereby preventing you from even getting infected or preventing you from even transmitting the disease. We were able to license this vaccine, and now we're trying to we're further studying this vaccine and trying to understand how best it curbs infection and transmission. But the key vaccine I would like to talk to you about today, which is widely administered, is in collaboration with the Indian government by the name of the ICMR, the Indian Council of Medical Research. This vaccine used an interesting platform. So you've heard of various platforms over the last 4 years. I'm going back to Vaccine 101 with a platform called inactivated vaccine. So this is a vaccine it's been used for decades, probably centuries as well, which involves you growing the virus in a large steel bioreactor, like a 100-liter, 300-liter steel tank, growing the virus at high concentrations, and then you kill it. You kill it with a chemical agent. So it essentially is completely, it's killed, it's inactivated, it has no ability to grow or infect a human cell, and then you use that as your vaccine antigen. This vaccine goes by the trade name of Covaxin, which was widely used in some of the global south countries. One of the key reasons why we were able to work on this vaccine is because it requires a highly contained facility. Think about the movies like Contagion, which I'm sure many of you have seen during the pandemic. High state-of-the-art manufacturing facilities where you have oxygen tanks on your back and you're breathing through a fully closed PPE suit. This is what we had essentially at Bharat Biotech, and we were able to grow live SARS-CoV-2 in these bioreactors, as we can see here depicted on this slide. That was one of the prime reasons why we were able to do it. So we had the resources and the skill set to work on these deadly viruses. But fast forward about a year's journey with Covaxin, we were able to do several preclinical trials. So these are trials done in animals to test initial efficacy and safety. The infrastructure in India is such so that it's fairly robust. So we're able to partner with various agencies to get these done. Clinical trials were also done in a rapid manner, phase 1 to phase 3 trials. And we're also able to test the vaccine's efficacy against many of these variants of concern over the years. And what's, I think, very important for South manufacturers to take note of is your ability to publish, get your data out there, and then help drive scientific conversation and help curb vaccine hesitancy. We knew this was a huge, burgeoning challenge, vaccine hesitancy. So we took it upon ourselves to ensure that we published our papers in very notable peer-reviewed journals. But Covaxin we were able to commercialize this product in about 12 months. So we're about 2-3 months behind Pfizer and Moderna. This was India's first vaccine grown through and through. So this was not with any collaboration outside of India. There was no technology transfer. There was no licensing. All of the IP was developed internally within the country at Bharat Biotech and the Indian Council of Medical Research. We were able to publish this vaccine, so it gives you a certain amount of validation that there is a need for such a vaccine. What we noticed, because this vaccine is very conventional, it's been there for decades, so the platform is very well established. We've not noticed any increased risk of myocarditis, pericarditis, or thromboembolism seen in a few other platforms. And over the last 4 years, we were able to supply 600 million doses of vaccines, primarily to India and also a few countries that are part of the global south. But I think, again, I mean, the picture I'm trying to paint is that all is not lost. I think in the first wave of COVID vaccines in 2021, 3 manufacturers came out of India. Serum Institute were making the Oxford AstraZeneca vaccine. The company that I work for, Bharat, was making Covaxin. And there was another collaboration between the University of Baylor and another Indian pharma company. But all in all, India was able to manufacture close to about 3 billion, 2.5 to 3 billion doses of vaccine. And as you can see by the chart over here, there is significant contributions coming from India, China, and South Korea. And what's failed to mention here is the contribution from the Brazilian companies and the Cuban companies as well. I'll just switch to a few other diseases. So just bear with me, another 2 or 3 slides, just to talk to you about the innovation that has happened pre-pandemic. Without that innovation, I don't think COVID would have been possible for us. And I'm speaking to you from the lens of being part of this larger consortium of the developing countries' vaccine manufacturers network. Meningitis A, a deadly disease, a high case fatality rate, led to around 30,000 deaths per year in the sub-Saharan belt in Africa. The Bill and Melinda Gates Foundation, in collaboration with my colleagues at the Serum Institute of India, another notable vaccine manufacturing company, were able to develop a novel meningitis A vaccine and roll this vaccine out all across this belt that you see on the map, and essentially have eradicated or eliminated meningitis A vaccine. This entire project took about 15 years in the making, and it was a novel vaccine. It also led to severe, the cost of this vaccine was only 40 cents a dose. So as you can see here, there's tremendous amount of cost saving, and the efficacy of this vaccine was close to 90%. Malaria is also another interesting antigen where we actually finally have 2 vaccines that have been approved. The first vaccine was approved in 2021. The second vaccine was approved in 2023. And I'm proud to say there are 2 companies working on this vaccine from the global south, of course, in collaboration, but there's somewhat of a global north and a global south collaboration here with malaria. Bharat, the company I work for, is collaborating with GSK for the first vaccine that was launched in 2021. And the second vaccine, by the name of R21, is in collaboration between the Serum Institute and Novavax and the Oxford University. Both these vaccines are being rolled out in Africa. This is where Plasmodium falciparum is the prime antigen of target. And as we go along this process, we believe that the yearly demand for this vaccine is about 50 to 70 million doses of vaccine. And the initial outreach, the input we've received from our customers in these countries is extremely high. So there's a huge amount of political will to get this vaccine introduced with a high coverage rate. I'd like to thank you for patiently listening to me. I'll be happy to answer any of your questions during the Q&A session.
Thank you so much, Dr. Ella. And just to remind everyone that yes, as the presentations are made, do post questions that you have in the Q&A section. After our third presentation, Dr. Vargas is going to speak for us now, and then we will have a discussant. I've introduced Dr. Wahid before. So now we will turn to Dr. Vargas.
Okay. Cool. We are going to look to mortality and life expectancy loss. Then I'm going to look into the global coverage. And then what is the impact of different vaccine technologies on contributing to the pandemic, and also at the end, the disparities in life saving across regions. There is many estimates about the death toll during the pandemic. but a recent article in the Lancet estimates that about 12 million deaths can be directly attributed to COVID, and an additional 4 million resulting from pandemic-related factors, for example, untreated cancer cases. The global average loss in life expectancy is about 1.6 years. Southeast Asia experienced the smallest reduction, 0.4, but Latin America and the Caribbean saw the most significant reduction of 3.6 years. This highlights the difference across the globe. In this graph, we have daily confirmed COVID deaths represented by the blue lines in the left axis, alongside the progress of the vaccines rollout represented by red on the right axis. In the year 2020, as you can see, with limited scientific knowledge, no effective treatments, and vaccine still under development, COVID deaths remained constant. But in 2021, there was a widespread transmission of the virus, which caused significant spikes, as you can see. The turning point came in December 2020 and January 21, when several vaccines became available. AstraZeneca, Pfizer, Moderna, Covaxin, Sputnik, and Sinovac. After this point, the pandemic started to recede, and by March 2022, the situation had improved. Over 360 vaccines projects were started globally. India and Latin America contributed with over a dozen each. Countries like Brazil, Argentina, Mexico, Chile, and Peru all initiated projects. But only half of those 360 made it to clinical trials. And from this set, only 25 reached phase 3. Finally, only a dozen vaccines received emergency authorization from WHO. Covaxin was approved in India in January 2021 and received WHO authorization by the end of the year. I think none of the Cuban vaccine received WHO authorization, but it was not clear to me from your presentation, Gerardo, and probably you can make a point later. Let's take a look at this chart. Show how vaccines have been developed throughout history. Each bar represents a disease and its timeline. The bar starts when scientists discover what caused the disease and ends when a vaccine is approved for that disease. Traditional vaccines took decades to be developed and completed. But in 2020, on top of the chart, everything changed. Scientists that year were able to develop several vaccines against this new virus in record time. Well, how this was possible? Well, we should not underestimate the power of focus. All the scientific community globally working simultaneously on vaccines and drugs instead of the usual fragmented focus of underfunded and coordinated work. The rapid and early virus genome sequence data that was deposited in a public platform like GSAID, the global initiative on sharing all influence data, was crucial. The genome provides fundamental insights into the virus, structure, and function, and that helps the development of vaccines and drugs. But the approved vaccines rest on decades of research. the messenger at least has 30 years of research behind. The same for the protein-based vaccines like the Cuban vaccine, and the inactivated viral vaccines about a century. So that's also another key point. Another important thing is the explosive production of knowledge. Sharing was crucial. An estimated 1.5 million articles were added to the global literature in 2020. Of these, in 2023, only 0.07% of those papers had been retracted, which means that they were of good quality. Also, governments and private organizations have allocated massive financial resources to combat the virus. And finally, the regulatory agencies adopted faster approval processes and clear guidelines about what a vaccine should look like following the flu model. Here I have data on coverage. Over 12 million doses were given globally. On December 30, 2022, 69% of the global population was at least had one dose. Of these, there are some, here is the average. India is above the average, and high-income countries are around 80% of the population have received one dose by that time. There are some countries like Cuba, Chile, China, Brazil above the average. We know that Cuba and China, they have their own vaccines. And Chile also was able to manage early contact with companies like Sinovac and Pfizer, so that the reason it has also a good coverage. Cuba got approved its vaccine in mid-21, so later than the others. But as you can see, this straight line, this vertical line, very quickly it was able to catch up and get to a super coverage, yeah? Lower income countries had, like Pakistan, had more than 60% of the population vaccinated. But in contrast, here, down here, low-income countries were just around 20%. So very low coverage, yeah? And now here I have a graph showing the doses by vaccine. So what is interesting is that, well, here in the top, we have Pfizer. Second, we have AstraZeneca. Third, we have Sinovac. And fourth, we have Sinopharm. Moderna is down here, and also here is Covaxin. If we add together all those doses, billions of doses, we get 40% of, around 40% of all vaccines count for the inactivated viral technology. Then about 33 is the messenger, the new technology with Pfizer and Moderna. And 25 is about the viral vector technology, where Oxford, AstraZeneca, Sputnik, CanSino, and Janssen's are. This graph doesn't have the protein-based type, where the Cuban vaccines fall. And first, the Novavax, that is the other protein-based vaccine, came very late in 22. So they didn't vaccinate many people with that vaccine. And I understand Cuba just was able to share the vaccine with a few countries. So what are the advantages? This is important. All the traditional well-established vaccines have not received much public attention, but they have many advantages. First, it's worth mentioning that those vaccines do not have patents for most, or at least strict patents. And that's, of course, a huge advantage because many manufacturers can produce them, yeah? Also, they don't require freezing, they don't require super freezing. And so they're an excellent option for health systems with limited resources, as they are very easy to store and to transport. And finally, as they were mentioned by the other panelists, they have been used on large scale, so there was time to refine them for decades, and they offer a very robust safety profile because, you know, rare adverse events only emerge with broader use. Finally, the COVID vaccine has been a game changer in the fight against the pandemic. A study by the Lancet estimates that the vaccines prevented between 14 and 20 million deaths globally in the first year alone. But the impact varies across regions. The most significant were in high-income countries, the purple, and in lower-income countries, green. India belongs to this category, so it's a major vaccine producer and distributor, and it plays a crucial role in this success. In contrast, upper middle income countries in blue, where many Latin American countries fall, did not do as well as you can see. Among the reasons were a lack of manufacturing facilities and vaccines arriving late. Finally, low-income countries saw the least impact. As you can see, yellow that is missing in this graph, yeah? So, in summary, some of the lessons learned, the sharing of data early in the pandemic was key. It's something that should be very important to support. Also, to foster more unified focus on some key health issues, since the fragmented and as usual type of research doesn't work very well. And that also will prevent resources from spreading thin into many projects. And finally, it's important advocate for global vaccine equity through the developing and sharing of vaccine technologies with non-prohibited patents, or better, with open source licenses. That's all from my side.
Thank you so very much, Dr. Vargas. Now, for my audience, we have had 3 wonderful presentations from our panelists. We will now go to our discussant, Osman Khalid Wahid. And I just want to remind you before he begins that remember, if you have any questions, please post them in the Q&A, because after the discussion, we will now have a time where you would have the opportunity to get the panelists to answer any questions you might have. So at this time, I'll turn to our discussant.
Thank you very much, Dr. Williams. And I'd like to congratulate all 3 panelists for 3 wonderful talks and 2 truly amazing success stories in Cuba and in India. I think there are lots of learnings from both these presentations and from Veronica's really lovely summation of the challenges and possible solutions. And I'd like to start the discussion with Gerardo. So Gerardo, first of all, congratulations on the truly excellent work that you are doing in relative isolation in Cuba. And I think the success or impact of those of your work, the health statistics really speak for themselves. I had the opportunity to visit your institute in 2007 as part of an international biotech conference. Just like Dr. Williams, I was really, really impressed by the amazing capacity that is there in terms of biotechnology and in terms of really cutting edge research. As you mentioned, and I think this is a point worth underlining, particularly for countries in the global south, that it is important to have capacity of production, but also the universality of the health system is critical. And that really showed in the graph that Veronica showed in terms of the quick coverage that you were able to achieve in Cuba. And this is a missing link in many countries, and I speak from one of those countries. In Pakistan, although we had a reasonable coverage by the end of 2021, really it took a very strong intervention, and it was a successful one in the end, where we had a national steering committee, which included members of the elected government, the military, and the bureaucracy, and a strong provincial coordination, and a campaign called RED, R-E-D, which stood for Reach Every Door. And that ultimately took coverage to much higher levels in the end, and I think it helped save a lot of lives. So one thing I think that really stood out from your presentation is that you showed really the WHO to be one of the challenges rather than an enabling mechanism. Now, of course, the WHO serves a very important role in the global health system in helping countries determine what is safe, effective, and appropriate to use, especially in times of emergency. And this is critical for developing countries where there is not enough local capacity to make those determinations. Yet, in your case, the WHO was a challenge. And in future pandemics, knowing what you know, what can the global health system, in particular the WHO, do differently? And what can you, as a major vaccine developer in Cuba, do differently so that this gap that we saw in the development of your vaccine and its availability in other parts of Latin America and the rest of the world, actually, where it may have played a very important role, was unfortunately not met?
Okay, so in our case, I think that it is important to be prepared in advance to fulfill all the requirements of WHO. In our case, because the pandemic, we use the facility that we already have for production of hepatitis B recombinant vaccine that was temporary, and we applied to WHO at that time. But as I mentioned, at the time that we applied, already I think the fast track established by WHO from the beginning was not the same. Pfizer vaccine was approved in 2 weeks. In our case, it was not like that. So we were to submit the document and we were waiting for answers. And later, so it took time. Later, as the facility that we have was a transitory facility, we decided to transfer the production of Abdala vaccine to a new facility that we built close to Havana. And it took time because at the time that you transfer your production to a new facility, that you need to validate the production in the new facility, you need to produce batches to show consistency, even to conduct some small clinical trials. So it took time. But for us, one difficult point was the qualification of that facility, because that was that we need foreign companies to qualify the equipment, to qualify the facility, and we needed to pay to those companies for that. And even having the money for that, the financial blockade that we have from the United States, it took several months to us to have the possibility to pay for that. It happened that most of the company, most of the, even the publishers have their accounts in American banks or in banks that follow the American rules. And they don't allow Cuba to transfer money. We cannot use those banks. And that is a big challenge for us because we need to deal with companies to try to find ways to pay through other banks. We need to ask the company to open accounts in different banks. They not each one is ready for that. And that is a complication. So it took several months for us to be able to qualify our equipment, our facility in a new facility, and to fulfill all the requirements. So we still are in the way. We didn't renounce to that. We still are in the way to qualify our vaccine by WHO, but we have many disadvantages for that. Meantime, we were able to register our vaccine in other countries. We mentioned, for example, in Mexico, we have been supplying more than 10 million doses to Mexico. So it's not mandatory to have the vaccine approved by WHO to be accepted by other regulatory agencies. That's why I mentioned that we need to homologize it. It's ongoing, an effort from Colombia, from Mexico, from different Latin American countries to homologize our regulatory system. I think that those efforts would be important to avoid this.
Thank you very much, Gerardo. And again, congratulations on overcoming some very serious concrete obstacles and yet succeeding in protecting the population of Cuba and of course, taking it outside Cuba as well. So I think what Gerardo's presentation very effectively highlights is the need for deeper South-South collaboration. And I'll turn now to Dr. Ella. Dr. Ella, as we saw in Gerardo's presentation, Cuba managed its vaccine programs with very meager resources. I think the total mentioned by Gerardo was about $30 million and an economic blockade. In comparison, Indian manufacturers like yourself and like Serum were able to forge very effective North-South alliances. Of course, you are already at a commercial scale. As you mentioned, 50% of global vaccines by volume are produced in India. So partnerships with the global North and your own domestic capacity, of course, help you accelerate your development and really help patients, not just in India, potential patients, not just in India, but also in several other countries. I also read that the Indian government at an early stage at least introduced or announced a $600 million grant to enable yourselves and Serum to scale up vaccine production, to accelerate development. And I'd like you just a little bit to spend a little bit of time talking about the importance of an enabling environment and how other countries in the global South can sort of connect with the capacity already in India and make more of a unified effort. You mentioned DCVM, and also it's a very important network. And how can such a network also help coordinate mutual recognition efforts at a global, at a regulatory level, which is another one of the challenges that Gerardo brought up?
Thanks for that question, Osman. I think global South, global North, I think overall collaboration is required for making vaccines. It's a highly complex pharmaceutical product. It is not as easy to make as a generic or a injectable pharmaceutical product in terms of complexity. In terms of the first question that you asked me is about the grant funding, the non-dilutive grant funding that exists, or the advanced market commitments that countries or customers need to provide us, I think play a pivotal importance in terms of financially de-risking projects for us. These clinical trials range anywhere between $50 to $150 million. The product development of the vaccine ranges between another $50 to $70 million. If you're able to get that capital at least upfront before a vaccine is even licensed, it will help you expedite those timelines. So you get to do many things in parallel. Now, I can only try to imagine the constraints in Cuba, as Dr. Gerardo mentioned, but at least in India, these constraints are also severely lacking, severely present. I mean, the percent of GDP spend on R&D is lacking. The government of India did come through with some amount of grant funding that were spread across different projects, but it's very hard to pick and choose which project will succeed. You look at Operation Warp Speed in the United States, they used a similar approach as well. They announced a huge fund, and they were able to give it to at least 5 to 8 manufacturers out there. Such a similar mechanism was in place in India, but I don't think it covered the full expense of product development and clinical trials. I think, at least from our perspective, I think it only contributed to 10% of the capital that you required to develop the vaccine. Many of these companies that you mentioned, Serum Institute, Bharat, Biologically Capable Indian Vaccine Manufacturing Network, capable Indian vaccine manufacturers, have their own resources to do product development and R&D on their own merit. And that's what essentially we did. There were some global north to south collaborations that we harnessed as well with the foundation and with CEPI that also helped de-risk it. But I think funding is one thing for a project. There is also something such as called an AMC, we call it in our industry, an advanced market commitment. So before a vaccine gets licensed, a manufacturer gets a quantum of money. And in case the vaccine is licensed, it reaches commercialization. That prepayment offsets the challenges of scale-up and R&D as well. So if you receive that capital before licensure, you get to start thinking about, how am I going to scale this up? I mentioned the steel tanks, the 300-liter bioreactors. If you got a prepayment, you could have probably done 1,000 liters or 5,000 liters. So you could have scaled up ahead of time. That, I think, was lacking in the global south because countries really did not have the balance sheets to go ahead and pick what vaccine could work and also give that prepayment as well. These are things that COVAX, Gavi are trying to address, and they did address to a certain extent, but I believe all of that was after licensure. So that tricky phase of product development, clinical trial, de-risking, I think is still lacking. And I think as a community overall, we need to improve that.
Thank you. And if you could also talk a little bit about DCVMN and other such networks in addressing this regulatory challenge, mutual recognition.
Sure. Mutual recognition is a tricky issue because it depends on the country of origin, where the vaccine comes from. It depends on that vaccine manufacturer, and it also depends on that regulator. I'll give you the Indian context. The Indian regulator is at a fairly mature level, not as mature as the FDA or the European medical agencies, but one notch lower. And if you get an Indian product approved with the Indian regulator, then other at maturity level, so you don't necessarily need a WHO recognition. Although it's important, it adds more weightage. At the end of the day, the pandemic was a seller's market. It depended on how much ever vaccine we produced, we found customers to take it. And for us, all that matters is that it goes into people's arms, regardless of which country, which NRA or regulatory agency needs to approve it. As long as it's being injected into people's arms and people are being prevented from infection and disease, I think we're okay with that outcome. But mutual recognition is something that I think needs to occur between two countries. Maybe bilateral treaties need to be a more focused agenda with certain of these countries that do not have vaccine infrastructure. And if they need to protect their citizens, perhaps they tie up with India, perhaps they tie up with China or Cuba to have that kind of mutual recognition, at least when it comes to the pharmaceutical landscape. But there is I agree with that point. I think that should be one of our action items moving forward.
Yeah, thank you, Dr. Ella. I think that was a very elaborate response, and you highlighted once again, first of all, the need for regulatory systems in manufacturing countries to be mature enough to be able to be mutually recognized by other countries. And also, the fact is that the WHO remains the gold standard for much of the developing world, particularly. And I think there needs to be a rethink also at that level on how it can be a more proactive and a more rapid response to developments that take place outside more traditional sources of vaccine production, and Cuba being a classic example. So Veronica, I'll turn now to you. I think one of the things you really highlighted very well, and it was present in both the other talks, was the fact that there are established platforms for vaccine development. And these established platforms are very safe. They have years, in some cases, over a century of safety data. And they're particularly important for countries in the global south because A, they don't have patents in many cases, and B, they don't require the more complex storage logistical challenges that, you know, the newer technologies like mRNA present, such as ultra cold storage conditions, and that really can make a huge difference in challenging logistical environments like many of the global south countries have. So what, in your view, is the role that academic partners like Harvard and the Mittal Institute play, and what should our next steps emerging from this very rich conversation today?
Okay, before answering that question, when I was putting together my presentation, what surprised me the most was how fast these vaccines were ready. I mean, I was really surprised about Covaxin being ready January 2021. At the same time, the Moderna and Pfizer. And I wanted to ask you, Ella, Dr. Ella, a little more, if you can elaborate how you were able to be there so fast. I know also that Cuba could have been ready by then if they didn't have all these hurdles, yeah? Because they were ready in June 21, so this is quite close, yeah? And they were able to vaccinate their population. Can you, Dr. Ella, elaborate a little more on this point? How you were able to get there so fast? You had already something ready and you just repurposed that? Please walk us through your journey.
Sure. I think it all starts with first, the essence or the ethos of the company. Our company is to innovate for the global south. So these are neglected diseases that nobody's working on, which we get to work on. So we have that privilege and that foresight in terms of our R&D acumen. So we have a skilled amount of colleagues at our companies that are able to tackle bacterial and viral pathogens. And we've already made about 5 innovator vaccines. This is with our own IP, 5 innovator vaccines that's being rolled out worldwide, and is contributing to Gavi and UNICEF as well. So having the vision, having the skilled expertise, having experience to various platforms. We've worked on viral vector, we've worked on inactivated subunit. We have experience juggling all these platforms. So when we started working on the pandemic, we were working on 4 different technologies, as I mentioned in my slides. We didn't know which will work, but we progressed with all 4 with equal importance to see which was getting to that clinical de-risking standpoint. And finally, I think it's the manufacturing ability that you have to not only product develop it, but also scale it up. And also at the same time, have that entire ecosystem around you, from your suppliers, your vendors who were able to get raw materials, who were able to procure the steel for the bioreactors. We have a very capable Indian FDA, the regulatory agency, which is at a certain maturity level, and they were able to do expedited reviews. We have access to a certain amount of grant funding and also access to labs that can do complex animal studies or complex microbiological assays. So I don't think credit only goes to manufacturers. It's very important to have that ecosystem around you. There's a big push for regionalization of vaccine manufacturing in certain parts of the world. The African continent is right now the main discussion point, which is a great initiative. I think it's very important for countries to have its own vaccine bandwidth, but the buck doesn't only stop with the manufacturer. You need to have these other pillars in place for you to be successful in the case of a pandemic or also for even routine immunization.
Another question, how many people do you have in your company working in this project, more or less? I will ask the same to the Cubans too.
Sure. So during the pandemic, we rapidly grew. We currently have around 4,000 colleagues at Bharat. I would say particular to this project, from R&D, product development, scale up of manufacturing, regulatory quality control, and packaging and distribution and supply, and the medical affairs team, close to about, I would say 350, 400 should be the number, somewhat involved in this project.
Okay, and another question from I show a slide with the explosion of literature, of papers published around the world. Do you have a system, a methodology to screen on those papers? Any of those one help you to faster your work? Can you elaborate on that?
Yeah, absolutely. I think, I mean, the pandemic was a time where everyone you would usually in academic settings, the mantra is publish or perish. I think that was followed in the pandemic as well. I think the first important paper was the genetic sequences of the virus that came out from a few Chinese investigators in January of 2020. Then we kind of knew that this was similar to SARS-CoV-1. So that was the precursor to SARS-CoV-2. Then it was fairly evident to us that once that paper came out of the genetic sequences, we identified the spike protein being the most immunodominant epitope, meaning that should be the target for the vaccine. There were also a few papers in the last 5 years pre-pandemic from the NIH and the University of Texas looking at structural biology, how to tweak these proteins and make them more intact and make them form a particular shape. It's called a pre-fusion spike conformation. That was important for us to develop several of our vaccines. And also there was a lot of guidance documents from, so these are not papers, research papers, but these are guidance papers from the FDA, EMA, India, and WHO, telling all vaccine manufacturers out there that, look, if you have a vaccine candidate, this is how we would like you to test it and evaluate it in animals and humans. This is how the quality control test should be done. So that kind of forward-looking guidance document was also very helpful for us, and it paved the highway for us to run on, essentially.
Excellent. Gerardo, Dr. Guillen, the same questions to you. I mean, probably if you didn't have all these issues, you will have been ready on January 21st. Do you think that's right? I'm exaggerating on around that this is correct.
You don't exaggerate. Of course, the recombinant platform is not so fast like mRNA or adenoviral vector or inactivated vaccine. It takes more time and its protein is different. So sometimes we have protein that is very difficult to purify and it takes longer to be able to solve the problem. In fact, it was not complicated in the case of RBD. Also, we designed a specific construction that facilitates the expression, the presentation of the antigen that allow us to patent the vaccine. That was important. But there are antigens that are more difficult than this to purify and to express. So that this technology takes time. But you're right. In this, we have not facing those difficulties. We have the vaccine. We will have the vaccine several months in advance. I wanted to add another well, answering your question also. We involve more than 200 people on this vaccine development. If you consider research, but production, quality control, quality assurance, so all this involve more than 200 people in our center. But I wanted to address another important point that I think that we should we should address that is related to the quality of the vaccine. So the quality of the vaccine is very important because that we are facing now, for example, in the United States, in Europe, almost half of the population didn't want to be immunized. And the problem with the side reaction, the mortality, the microcoagulation problems that happen with the mRNA vaccine and also with adenoviral vaccine from AstraZeneca recently. So this damaged a lot the confidence of the population about vaccine. And that is a big problem. We have not that problem in Cuba because our platform is very safe, the quality of the vaccine is very high, but we need to be careful when we are using a different platform and how we design our vaccine. But because we are doing nothing, if we are developing the vaccine, but the population don't want to take the shot. So this is another point that we should be careful about.
Yeah, I think this is a point that actually nobody has done so far, is that in developing countries, in India and in Latin America, the acceptance rate was quite high. We didn't have the problem that the US had with the anti-vax. And one of the reasons probably was because those were very traditional technologies that were highly accepted. I do have another question regarding the protein-based technology. I work on innovation, and in Latin America, this is the technology that most scientists use in the last decade. But I'm really surprised why this technology is so underrepresented in the total number of vaccines, because the ones that are really strong were the inactivated viral, the messenger, and then the viral vector. But the protein-based is really underrepresented. Any thoughts on that?
Yes, my opinion, what I was mentioning, that this, if you have the mRNA platform or adenoviral platform, you can introduce your cassette, that codified for your antigen, and you always, with very high percentage of success, you will have your vaccine. But in the case of a recombinant platform, you cannot assure that in advance. So each protein is different. You cannot extrapolate one purification process to another. So the way we are purifying our antigen for COVID vaccine is completely different than the way we use to purify our antigen for hepatitis B vaccine. So each protein is different, the glycosylation, the three-dimensional structure of the protein. So it's complicated to purify protein. So that is why other technologies could be more appropriate for pandemics, that the recombinant protein. In this case, we were successful, but we cannot assure that with every antigen, we will have the same rate of success. So I think that is why other companies select other platforms for the development of the vaccine for pandemic. And you always should have in account the risk-benefit balance. So in the case of pandemic, you can risk more on safety because you will save millions of people immunizing those people. But when you have not epidemic in front, when you have not the pandemic, it's not the same. You need to think about the risk, and you need to be more careful. That's why I'm not support so much the tendency that is now to use the mRNA platform for everything. So you can use mRNA platform, for example, for therapeutic vaccines, because the benefit will always in favor. But for prophylactic vaccine, and especially for children, you need to be more careful about the technology that you will select.
Thanks.
I'm watching the time. Our time is ticking away very quickly. We want to make sure that our moderator discussant has time for any other issues he wanted to raise. And I see one question in the chat, and we want to get to that. But let me go back to Osman.
Thank you, Dr. Williams. And I was really enjoying this back and forth. So thank you, Veronica, for asking those questions. So one comment I did want to make, and it relates, in fact, to the question I'm going to read out, is Rachel pointed out a very important enabler for vaccine development, and that is de-risking the process. Particularly if you want to see more vaccine development in the global south, you really want to avoid market failures, particularly in countries where development is to be done by the private sector and not by a state-owned company. Mechanisms like COVAX and bilateral donations very clearly saved a lot of lives in the process of COVID, but also in the absence of domestic government commitments to purchase locally produced vaccines or any large substantial grant, it also in a way suppressed any local development. And I think in the context of resilience and in the context of the importance of diversifying the global manufacturing network, I think these, in fact, perhaps unintentionally also serve as barriers. So the question I'll pose to the panelists is related to that a little bit. It's, what are your thoughts on COVAX? And this is a question by Prerna Makkar. I hope I pronounced the name correctly. Rachel, would you like to go first?
Sure, Osman. I think, she asked a very valid question, and Osman, I think you have answered and summarized it to the T. I would say in terms of de-risking, in terms of if we are to work on a HIV vaccine, hypothetically, what would we need? The first thing we would need is some kind of collaboration to work on a vaccine. The second is some kind of grant funding that needs to be provided by a large philanthropic organization or a donor like CEPI. The third would be to have some foresight as to the commercial viability of this product if it reaches the market. And I think that's where COVAX plays a pivotal role. It gives that confidence to manufacturers and developers that yes, you can work on a vaccine. And if you meet our guidelines of how the vaccine, well, their only guideline is it has to be WHO approved. So once if it meets WHO regulation, we will buy it. Now, how much we will buy, how long we will buy for, those metrics are also defined by COVAX, which they did define for the pandemic as well. What COVAX helps Global South manufacturers is a one-stop shop solution with regards to vaccine distribution and supply. So instead of us talking to 150 countries, 150 Ministry of Health in different parts of the world, we just need to liaise with COVAX. The invoicing happens through COVAX and then they tell us where to ship it to. So COVAX is, I think a remarkable achievement which happened during the pandemic. I know that the allocation of vaccine wasn't up to that promised goal. That was because of several other reasons of vaccine nationalism, which is another issue we need to curb moving forward. But I think the platform is set for a unified procurement agency to be the central voice for upcoming pandemics.
Thank you. Gerardo, would you like to respond?
I think that this is not only the vaccine. For vaccine, of course, we need financial support, but it should come for international organization because private companies will not risk. Private companies don't want to risk in so complicated projects, and they want to have incomes more quick. So it's very difficult to get money from companies to invest on vaccine development and to invest on innovation. So we need, for example, the Bank of the South, the organization like CELAC in South America. We need the BRICS, for example, the BRICS Bank, the Chinese initiative of the Silks and Root. So those organizations are the ones that I think could give support for this effort. But the effort should not be given only to build the capacity for production and develop the vaccine, but also the infrastructure to administer the vaccine. Because we knew, for example, that some countries in Africa, they received 1 million of doses from COVAX, and they were unavailable to administer the vaccine to the population because they had not the infrastructure to distribute and to administer the vaccine. So that is another important point, to prepare the human resources for that, to create the infrastructure, to create the cold chains. That was, for example, an advantage of our vaccine, on our platform, and also an activated vaccine, for example, that was not the same requirement that mRNA vaccine, that they needed freezing cold chain. That was not affordable at all, was not possible. That infrastructure didn't exist in the South Cone. So those cones are also important where we are talking about to be suitable with the impact of our vaccine.
Thank you very much for that comprehensive response. And I think given the shortage of time, I'll now turn it over back to Dr. Williams for his summation.
Thank you so much. And thank you to our audience for listening to this really fascinating and instructive conversation we've had today. Before I turn to the vote of thanks, I want to say 4 points that I think I learned today is the importance of a universal health system when addressing a major national and global public health channel. Another point I learned today was the importance of large regulatory agencies or large international entities like the WHO being an enabler instead of an obstacle when dealing with public health crises. A third point I learned today is the adverse impact of the US embargo of Cuba and how it hurts the public health infrastructure of Cuba. And as I am personally a child of the Caribbean, when you hurt Cuba, you hurt the entire region. Cuba is the largest country in the Caribbean area, and it is a good big brother, providing medical professionals throughout the region, providing scholarships to many young people in much poorer countries who couldn't go to medical school, and Cuba provides that. So when America hurts Cuba, it hurts the entire Caribbean and Latin American region. And my final point is we live in a global village. We are all connected, and we need to see what we can do to work to improve the health of all. With those final comments, I turn to Hitesh Hathi, who will offer our closing vote of thanks.
David, thank you very much. That is so well put. It's very difficult to follow, but I think you're talking about a global village is exactly right. We are in a global village and all of us are connected. It's something that we know, and yet somehow we fall short of building the structures to realize, the full potential of the reality that is there. I also agree with you, your summation, perfect. And I think we all also arrived at within every country and beyond borders of countries, we need structures in place to make possible the kind of public health that we all seek. Listen, I'm not an expert on healthcare or medicine, and yet I have to say I was riveted for this entire time. I was very taken by the perspectives, David, from your large overview, which thinks deeply about some critical issues, to each of you, from the actual manufacture and research and development to, so Usman, you actually are in pharmaceuticals, Veronica, you've brought the larger global health, economics, and also kind of research perspective. It's a very comprehensive and interesting set of looks into this issue. I'm very grateful to all of you. I'm very grateful to our audience for listening. We will be doing more of this, and I want to finally end with a vote of thanks, not just to you, but also to the team at the Mittal Institute, which worked very hard to put all of this together. Thank you. And I want to thank our partners, especially the Rockefeller Center for Latin American Studies here at Harvard, the T.H. Chan School of Public Health, also at Harvard, and the Lancet Citizens Commission, which is part of the Mittal Institute. And David, to your last point, the Citizens Commission is in fact charting out a path for universal healthcare for India and South Asia, and we will follow with more on that. So thank you very much. Good day, good evening, good morning, wherever you are in the world.
Thank you, thank you, thank you. Thank you all the panelists. This has been absolutely outstanding.
Thank you. Thank you.
Thank you.
Enjoy it.